Welcome to Complementation Test for Dummies.
So what are complementation tests? Complementation tests are used to determine if mutations are on the same gene. It's important to note that complementation tests must be done for haplosufficient genes. A haplosufficient gene only requires one wild-type copy to display a wild-type phenotype. Haplosufficient literally meaning one is enough. For example, if one copy of the gene were to be deleted or mutated, the resulting phenotype would still be wild-type. Conducting the test is very simple. First, you induce mutations. Then, you separate the mutant organisms from the wild-type organisms. The test cross occurs when you cross each mutant with every other mutant. Upon examining the F1 progeny, you should be able to determine if the parental mutations are on the same genes or on different genes. If you observe a mutant phenotype, then you know the mutations are on the same gene, gene 2 in the example to the left. This is because both copies of gene 2 will be mutant. Conversely, if you observe a wild-type phenotype, then you know that the parental mutations were on different genes. The offspring then has one wild-type allele and one mutant allele for each gene, as depicted on the right. As we discussed before, since the gene is haplosufficient, the singular wild-type will be enough to result in a wild-type phenotype.
So why yeast? Yeast is a great organism to conduct complementation tests with for several reasons. To begin, it is a simple, single-celled eukaryotic organism, meaning its cellular contents are the same as those seen in humans and other animals. This means that the genetic observations in yeast are likely to be true in these non-model organisms. Yeast is also very easy to culture, can be found in both haploid and diploid states, and the adenine synthesis pathway, the pathway we will conduct our complementation tests with, has already been well researched and all known enzymes in the pathway have been documented. This pathway can be seen to the right, and our complementation test will focus on the red-colored 81 and 82 enzymes. Without both, yeast could not produce adenine and instead produce red pigment. Below is a reproductive cycle for yeast, showing how it moves from both haploid and diploid phases. This is beneficial for us as we can see recessive mutations with their phenotypes present in both the haploid and diploid phase, which will be crucial when we plate our samples. In order to induce mutations in our yeast, we applied UV radiation to the colonies using our yeasty bake oven, a state-of-the-art complementation testing device found only in the Hartman lab at Yale University. The exposure to UV radiation forms pyrimidine dimers in the DNA, which if not corrected by DNA damage repair mechanisms, will result in the introduction of mutations in the genome of our colonies.
Then, we isolate the mutant phenotype yeast across all possible mating variations and then cross them with each other. You'll notice that we only cross A with B, because only crosses between A and alpha will produce diploid offspring, which is essential to the complementation test. Remember, red colonies are mutant and a result of parental mutations on the same gene, and white colonies are the result of mutations on different genes.
Here we take a deeper look at the complementation test results at the molecular level. On the left, we see the cross between HA2 and HB2, in which both crossed haploid mutants have a mutant 82. This results in diploid progeny with the mutant phenotype. Conversely, on the right, we see the cross between HA1 and HB2, in which one of the haploid mutants has the 81 gene mutated and the other has the 82 gene mutated. When crossed, these genes complement each other and allow for a wild-type phenotype. And that's all. Now you're an expert in complementation testing.